Cells were first described by the English microscopist Robert Hooke in 1665. Here we see his illustration of cork with cells, which he described as filled with juices and by degrees sweating them out. In 1781, Fontana in Italy described in these slime cells from the eel an oval body furnished with a spot in the middle. This spot is generally taken as the first description of the nucleolus, although he did not draw it. The term nucleus was coined by Robert Brown. In 1838, Schleiden published his classical work, Contributions to Phytogenesis, and some cells, which he called cytoblasts and sporules, can be seen in this illustration. It's obvious that the membrane, the nuclei and nucleoli had been clearly recognized by the middle of the 19th century. Although Corti in 1774 was the first to describe streaming, the fine arrows in this potato hair cell of Schleiden's are probably the first illustration of streaming in the literature. In 1890, Altman, whose frog liver cells are seen here, gave the name Altman's granules to what we now call mitochondria. Towards the end of the 19th century, there was much discussion about whether fibrils in the cytoplasm were artifacts or not. In Italy, Golgi detected the Golgi body with his newly developed silver stain. In Golgi's original illustration, it can be seen permeating throughout the cytoplasm. This view from a paper of Holmgren in 1902 also shows the Golgi apparatus distributed rather differently in the cytoplasm, that is, in the area around the nucleus. The next structure seen by the light microscope was the nucleolonema, detected by Estebel and Sotillo in Uruguay in 1950. They gave this name to the fine fibrillar structure apparent within the nucleolus. Recently, under light microscopy, a nucleolar membrane was seen in all neurons examined from frog, guinea pig, rabbit and rat. Initially detected by phase contrast microscopy, it has now also been seen with magnifications as low as 200 times with bright field, dark ground, interference, polarizing and anopteral microscopy. However, the cells must be dissected out in saline in order to see it. It is not known whether the membrane appears around the nucleoli of cells other than neurons. The electron microscope, which was applied to the study of biological tissues in the 1940s, permits magnifications 2 to 3 orders greater than is possible by light microscopy. As a result of observations of tissues with this greater magnification, a number of new structures were seen. These are illustrated in this plasma cell from Bloom and Fawcett's classical textbook of histology. The new structures are the endoplasmic reticulum, first seen by Porter, Claude and Fulham, the nuclear pores by Callan and Tomlin, and the unit membrane by Robertson. The existence of the Golgi body, about which there had been much controversy up to the middle 1950s, was regarded as having been proved when it was seen on electron microscopy as in this typical picture of Toner and Carr. Christie was seen for the first time in mitochondria. This well-known diagram by Brachet of the generalized cell is one of two which appeared in the Scientific American and have been widely reproduced in textbooks. In it, the following features can be seen. Within the cytoplasm there is a Golgi body and an extensive endoplasmic reticulum which is connected to the extracellular space and to the nucleus. Pores are present in the nuclear membrane. Here is the other diagram by Robertson. It shows all the features of the Brachet cell, but in this one the membranes all appear as double lines, and therefore there are cisternae between them. Both diagrams are intended to show the living cell, and both are regarded as being representative of plant as well as of animal cells. Let us now have a look at some of the features of living cells. Streaming in plant cells is often one of the first features to be observed under the microscope by biology students. It can be seen here at low magnification and in real time. In mammalian tissue cultures, it is possible to see several kinds of intracellular movement. Phagocytosis, here speeded up 96 times, is occurring in a macrophage. Large particles are engulfed by the cell and move around the cytoplasm in vacuoles. Brownian movement of bacteria can be seen in the cytoplasm. Pinocytosis is a similar process. There is a small active cell in the upper half of the field, but we are interested in the large chick embryo cell occupying most of the lower two-thirds of the screen. Vacuoles of fluid are engulfed, particularly near the arrow, and can be seen moving in the cytoplasm. This sequence is speeded up 48 times. Continuous movement of mitochondria is also a feature of the living cell, seen very clearly in this time-lapse film. Brownian movement of small particles is also seen here. Nuclear rotation is illustrated by this cell from a culture of rat hearts speeded up 48 times. The complete list of intracellular movements includes diffusion of injected particles, Brownian movement, phagocytosis, pinocytosis, movements of mitochondria, formation of vacuoles and their disappearance, and nuclear rotation. These can be seen in living plant cells, protozoa, or cells in tissue culture, and are the main criteria by which one judges whether the cells are alive. When intracellular movements stop, the cells are dead. The purpose of this film is to arrive at the real structure of the living cell. We shall examine the generally accepted structure seen by electron microscopy in the light of what is known about, firstly, the properties of the living cell, which we have just demonstrated, secondly, the limitations of solid geometry, and thirdly, the physical properties of materials. Let us consider the endoplasmic reticulum. The first question that arises is, how is it possible for the intracellular movements which we have seen in living cells under relatively low power light microscopy to occur if there are indeed a three-dimensional net which requires 100 to 1,000 times greater magnification to see and, according to modern textbooks, is attached to the cell membrane and to the nuclear membrane? This point is illustrated by examination of some intracellular dimensions. The distance between the strands of the reticulum in a number of preparations varies between 800 and 2,000 angstrom units, but mitochondria which appear at least 5,000 angstrom units wide are seen to move freely, and particles up to 50,000 angstroms can be seen in Brownian motion. Of course, the reticulum does not completely fill up the cytoplasm, but the rate of streaming and Brownian movement will be determined by the narrowest space between the reticulum and the cell membrane, which would act as a bottleneck. The next question is, how is the reticulum attached to the cell membrane? According to the widely accepted unit membrane concept of Robertson, on electron microscopy, the cell membrane and the endoplasmic reticulum both appear as two lines with a narrow space between them. If the two layers of the cell membrane invaginate to form the reticulum, as Robertson has suggested, the reticulum should always appear as four lines, or cisternae should always be seen when the reticulum is present. This would give us one model of the attachment of the reticulum to the cell membrane. There are two other possible models. Each layer of the reticulum could be attached to the inner layer of the cell membrane, and the outer layer would be imperforate. This is occasionally seen, but would necessarily mean that the extracellular space could not be continuous with the channel in the reticulum. Alternatively, the lumen of the reticulum could make a hole or cleft between both of the two lines of the unit membrane. Thus, one could see holes in the cell membrane at the edge of which the two lines would stop abruptly. Unfortunately, the latter appearances seem not to have been seen on electron micrographs at all, and the cisternae associated with the first model are difficult to find. Indeed, in an extensive examination of the literature, one rarely comes across electron micrographs, as opposed to diagrams, showing the point of attachment of the endoplasmic reticulum to the cell membrane. Our next difficult question is, what is the geometry of the reticulum and indeed all other unit membranes? This model represents a unit membrane. Let us say that it has a total thickness in life of 150 angstrom units. We can cut transverse and oblique sections. As the sections become more and more oblique, the two lines of the unit membrane will appear farther apart. So, we would expect to see a range of distances between the two layers of a unit membrane of a particular thickness, depending upon the obliqueness of the section. It would be impossible that the unit membrane would always appear to have a constant thickness. In the present context, this applies to the cell membrane, the mitochondrial membranes, the cristae of the mitochondria, and the lamellae of the Golgi body. But it is also a serious question in relation to the myelin sheath, the synaptic cleft, and the lamellae of the chloroplasts. It would be quite inconceivable that all electron microscopists always cut perfect transverse sections of all subcellular organelles which all happen to be concentrically orientated simultaneously. All the structures in these three adjacent cells could scarcely be expected to have been in the same orientation at the time of cutting the section. It is equally surprising that in the literature the distances between the two lines of the unit membrane, the nuclear membrane, and the cristae are normally given in a particular tissue as one figure without a range. Electron microscopists have two views on the form in 3 dimensions of the endoplasmic reticulum. According to one view, the endoplasmic reticulum consists of flattened vesicles or sheets. If we look at it in transverse section, we see the traditional equally spaced parallel lines with a few connecting walls. In this orientation, we see a solid sheet of tissue wider than the distance between the sheets. It should be noticed that in this orientation, the underlying sheets must appear closer together than they do in the original transverse section. The second structure proposed for the endoplasmic reticulum is of a real net, as the name implies. Looking from above at this model, we see the strands of the reticulum in several planes. In different orientations, we get a further series of geometrical patterns. Nevertheless, on transverse section, it will appear again as equally spaced parallel lines, although connecting strands will also be seen. Unfortunately, shapes other than the well-known parallel lines are hardly ever seen. We may summarize these geometrical arguments by saying that the endoplasmic reticulum is hardly ever seen as a section of a possible 3-dimensional structure. It exists only in the 2 dimensions of the picture. In this respect, the endoplasmic reticulum should be contrasted with the whole mitochondria and the muscle filaments, which do appear as 3-dimensional structures on electron micrographs. Put another way, it would be quite impossible to construct a 3-dimensional model of a whole cell based on an electron micrograph, which would allow the currently accepted intracellular movements to occur. Let us now look at another structure seen by electron microscopy, the nuclear pore. Nuclear pores were originally noted as complete gaps in the nuclear membrane, appearing as circles on tangential sections as Lanka and Scheer have photographed them, or as slits as seen in this typical picture. More recently, they have been described as being closed by a diaphragm or fenestration. Elaborate drawings have been made, like this one of thread goals. Note that his pore diameter is between 500 and 520 angstrom units. Pore diameters in the literature vary from a maximum of 1,000 angstrom units in rat cerebellar cells to a minimum of 200 angstroms in mouse pancreatic acinar cells. More recently, smaller values have been cited since the description of a nuclear pore apparatus. Let us now look at the size of some ions and molecules found to be either side of the nuclear membrane. Their diameters do not exceed 59 angstrom units for chlorophyll, but molecules less than 10,000 molecular weight are less than 35 angstroms wide. How could such large pores prevent the ingress of such small ions or particles? Furthermore, the pores are said to occupy between 3 and 32% of the surface of the nuclear membrane, and it would require a phenomenal amount of energy to prevent molecules leaking through such a relatively large area of holes. Lowenstein and his colleagues have reported that there are potential differences across nuclear membranes of nearly all the nuclei they have examined. Would not such potential differences be short-circuited by these holes? One may also ask how nuclear rotation could occur if the layers of the nuclear membrane were riveted by the nuclear pore apparatus shown in Threadgold's diagram. This diagram also illustrates a geometrical difficulty related to nuclear pores. We have seen them on electron micrographs as slits or circles. Why do they never appear oval? They should sometimes look like indentations on a golf ball, as illustrated here. A further difficulty arises when we consider a section of a hole in the membrane. Here we see a model of a pore face on. This is the width of the section. It is quite evident that the pore cannot be seen in transverse section if the diameter is less than the thickness of the section. This is true even if the section cuts one face of the pore. Only if the pore has a larger diameter than the thickness of the section and is cut both posteriorly and anteriorly by it will it be seen at all. The size of the pore will apparently be that of the smallest gap. Obviously, the size of the gap appears will depend on where the hole is in the section. Therefore, we should not expect to see a constant pore size. The circular shape of the pores on plan view leads us to suggest that they are bubbles, and on transverse section they represent cracks in the nuclear membrane. At this point, it would be useful to put forward our suggestions about how the endoplasmic reticulum, the unit membrane, and the nuclear pore originate during preparation for electron microscopy. If we look at this illustration from Weekly's Introduction to Electron Microscopy, we see a section unstained on the left and stained with heavy metals on the right. Obviously, the image the electron microscopist sees is composed only of heavy metal. Furthermore, the two-dimensional appearance of the structures we have been discussing gives us a clue as to their genesis. That is, they must be formed after the sections have been cut. A geometrical line is a fiction because it has position but no thickness. Any real layer has two surfaces. This layer represents a real membrane. Heavy metals like osmium, tungsten and lead are deposited on surfaces of such membranes for electron microscopy. And as we see here, the membrane appears as two parallel lines of heavy metal. Thus, any stain which is a deposit like a heavy metal will never permit us to see a real membrane as one line. This point applies equally to freezing techniques. Although freezing is used for fixation, heavy metals are also used for seeing the tissue. When a tissue is prepared for the electron microscope, it contains some embedding medium, some heavy metal, and possibly some tissue. Each of these three components is grossly different with respect to its coefficient of expansion, heat conductivity, affinity for metal, electron density, stability, vapor pressure, and compressibility. Let us now look at one of these, the temperature coefficient. The figure for osmium is approximately 1/10 of that of the epoxy resins, and the temperature inside the electron microscope specimen has been measured and is several hundred degrees. Therefore, the tissue, the metal, and the embedding medium, with their vastly different coefficients of expansion, will explode if rapidly heated to high temperature, as does a piece of clay in a potter's oven if it contains air bubbles. There is no doubt that on electron micrographs one can see the appearance of an endoplasmic reticulum in the cytoplasm of most cells, so that if one believes it to be an artifact, one should be able to explain it. We can describe the cytoplasm in a living cell as an aqueous suspension containing, among other constituents, salts, amino acids, fatty acids, and many metabolites soluble in water. When the tissue is dehydrated and organic solvents are added for electron microscopy, these solutes must precipitate and insoluble particles will deposit. The distinguished cryobiologist Luyet and his school in Madison have shown the beautiful patterns which may be obtained when various solutions are frozen and then viewed with the electron microscope. These are the patterns made by freezing salts, and these made by freezing amino acids and glycerol. When freezing to below minus 50 degrees occurs, ice crystallizes out and the solute precipitate. However, it is most significant that the particular pattern found depends upon the rate of freezing, the nature of the solutes, the purity of the constituents, and their relative and total concentrations. It is of particular interest that careful examination of their precipitates by electron microscopy often reveals the two-line thick appearance, and these two lines appear to be of a remarkably uniform distance apart. Is this a model for the unit membrane? Finally, let us go back to an intracellular structure that predates the electron microscope. The Golgi apparatus appeared to Golgi as a uniform net distributed within the cytoplasm. To later light microscopists like Born, it appeared as a paranuclear mass of about the same total diameter as that of the nucleus, and to Holmgren it was a shell of granules spherically distributed around the nucleus. The controversy as to whether the Golgi apparatus was real or was a fixation or staining artifact was considered to have been settled when the body was seen by the electron microscope. This is a typical picture of its lamellar structure, and its appearance at this magnification is extremely uniform throughout all tissues. However, when we compare the appearance and dimensions of the Golgi apparatus, firstly as seen by Golgi, secondly by the later light microscopists, and thirdly by the electron microscopists, it becomes abundantly clear that they were looking at quite different objects. For example, Golgi's network looks nothing like the electron microscopist's onion-like lamellar structure. The paranuclear Golgi apparatus is never less than 8 to 10 microns in diameter, while the electron microscopist is never more than 2 microns. Although early phase contrast microscopists like Ludford claim to have seen the Golgi apparatus in unstained preparations, it must have been several orders larger than the lamellar structure seen by the electron microscopists. We can now reconsider the structure of the living cell in the light of the present discussion, after having shaved off unnecessary hypotheses by the unsparing use of Occam's razor. All intracellular structures are moving continuously and at different speeds. If we were to stop this movement and draw the generalized cell, it would look like this diagram. The membranes around the cell, the nucleus, the mitochondria, and probably the nucleolus are of single thickness and are imperforate. There is no endoplasmic reticulum or Golgi apparatus. The lysosomes have not been considered in this discussion. The nucleolonema is present in the nucleolus. Unhappily, it is a picture remarkably like that which had been revealed by the 1890s, with the sole exception of the further detail of the nucleolus. The lessons that come out of these considerations are, firstly, that we should always assess the validity of any structural findings in cells by their compatibility with observations on living tissue. And, secondly, that we should pay much more attention to the possible, indeed probable, effects of our preparative techniques upon the appearance of cells.